Hey everyone, and welcome to this deep dive where we're going to be tackling, something a little different today.
Yeah,
Animal models in tuberculosis research.
it's a good one.
It might sound kind of dry at first, but trust me, this is going to be fascinating. We've got a whole stack of scientific papers and reviews here.
Oh, yeah.
Some going back as far as like 2009.
Wow.
Some hot off the press from this year.
Right.
And we're going to really try to get into, how scientists are using animals to study tuberculosis.
Right.
What are we learning from these models?
Yeah.
And what are some of the limitations?
And of course, the ethics.
The ethics, of course, a big one. So, it really makes you think, how closely can we actually mimic human disease in an animal?
Yeah, that's the big question.
And what are the ethical considerations there?
For sure.
So-
So it can be a good one.
I think it's going to be really interesting. So, first things first, why even use animals to study TB?
Well, studying tuberculosis directly in humans has some pretty major limitations.
Yeah, I can admit.
It's a disease that can take years to develop.
Right.
And we can't ethically, expose people to a potentially deadly pathogen.
Right.
We can't just do that.
Yeah, of course not.
So, and even then, getting detailed tissue samples throughout that whole process would be almost impossible.
Yeah, that makes sense.
So animal models really give us this controlled environment
Right.
where we can study how TB unfolds.
It's like a zoomed-in view of the battleground,
Exactly.
You could see the TB bacteria and our immune system going head-to-head.
Yeah.
I'm hooked already.
It's fascinating.
So what kind of animals are we even talking about here?
Wow.
Our sources mention quite a few different species.
Yeah, there's a bunch.
Yeah.
I mean, mice are definitely the real MVPs of TB research.
Okay.
They're small, they're affordable, and we can even tweak their genes to study specific aspects of the disease.
That's incredible.
But of course, no model is perfect.
Right.
One big difference is in those, granulomas,
The granulomas.
those little fortresses that form to contain the infection.
Right.
Mouse granulomas are missing some key features.
Yeah, like necrosis, right?
Yes, exactly.
Where the cells basically self-destruct as defense mechanism.
Yes, wow.
And hypoxia,
Yeah,
which is a lack of oxygen in the tissues.
it gets intense.
It sounds intense.
It is.
So that's where other animal models come in,
Yeah.
like guinea pigs.
Guinea pigs, yeah, they develop those human-like necrotic granulomas.
Okay.
So it gives us a more accurate picture of what's happening in our own body.
So it's all about choosing the right model for the job.
Yeah,
It's like having a toolbox
correct.
with different tools for different tasks.
Exactly. Now, rabbits.
Rabbits.
Rabbits bring something totally unique to the table.
Okay.
They can develop cavitary disease,
Okay.
which is a more severe form of TB where actual cavities form in the lungs.
Oh, wow.
Yeah. So this is crucial for studying that specific aspect of the disease.
Okay, so mice, guinea pigs, rabbits.
Mm-hmm.
Now this is where it gets really interesting.
It does. Monkeys.
Yeah.
Or to be more precise, non-human primates.
Yeah.
They're obviously closely related to us,
Of course.
which makes them valuable for research, but
Yeah, but
big ethical consideration.
ethical considerations for sure.
And even with the NHPs,
Yeah.
it's important to remember that they still don't perfectly mirror
Right.
human TB.
Right, they don't.
There are subtle differences in how the disease progresses.
For sure.
So it's all about finding that balance between the scientific value of a model
Yeah,
and the ethical implications.
absolutely.
And then we have a wild card,
We do.
zebrafish.
Zebrafish.
I'm picturing them with a tiny little stethoscope in a tiny tank.
Right. I know,
Yes.
it's funny. But because zebrafish embryos are transparent,
Oh, what?
scientists can actually watch the infection happen in real time.
Oh, that's incredible.
Especially those early events like granuloma formation.
Wow.
Of course, they're not suitable for studying the full course of the disease.
Right.
But so we've got this whole range of animal models,
Yeah, we do.
each with its own strengths and limitations.
Absolutely.
So how do scientists even decide which one to use?
Right.
It seems like a tough decision.
It is. It's a balancing act.
Yeah.
They have to consider the specific research question, the complexity of the model, the cost, and of course those ethical considerations.
Right.
Sometimes a simpler model, like a mouse, is enough,
Okay.
and other times a more complex model like rabbits or NHPs is necessary.
It's like assembling a team of experts.
Totally.
Each with their own unique skills to tackle this complex problem.
I love that.
So what have we actually learned
Right.
from all this research using animal models?
Yeah.
Our sources highlight some pretty crucial insights into how TB works, how we develop new treatments, and how we test potential vaccines.
Yeah, let's start with understanding how TB actually develops in the body.
Okay.
the pathogenesis.
Okay.
Animal models have been absolutely critical in figuring out how Mycobacterium tuberculosis, the
Right.
bacteria that causes TB, infects different cells in our bodies.
Especially those alveolar macrophages, right?
Yeah.
Those immune cells in our lungs that are like the first line of defense
Exactly.
when we breathe in TB bacteria.
That's exactly right.
Okay.
Animal studies have revealed how those macrophages and other cells get infected
Right.
and how TB manages to, gain a foothold in our lungs.
Yeah.
We've also learned a ton about the role of those granulomas,
Right.
those little fortresses that try to wall off the bacteria.
But sometimes the bacteria can escape or even use the granulomas to their advantage, right?
They can't, they're sneaky.
They're sneaky little bacteria.
They're so resourceful.
Yeah.
Animal models have allowed us to study this intricate dance between the immune system and the bacteria.
Right.
And we now know that M. tuberculosis has some pretty impressive evasion tactics, allowing it to hide from the immune system and persist in the body for years, sometimes even decades.
Right.
That's what we call latent TB.
It's like a tiny time bomb ticking away in your body. It's not a pleasant thought.
Not a pleasant thought, but understanding these mechanisms is crucial for developing effective treatments and vaccines.
Absolutely.
So speaking of treatments,
Yeah.
animal models are essential for testing the effectiveness and safety of new TB drugs
Right.
before they ever reach human trials.
Exactly. It's like a dress rehearsal
Okay.
for before the big show.
Okay, I like that.
Scientists can test different drug combinations, dosages, and treatment lengths in animals to figure out what works best and if there are any side effects.
Right.
And this research has already led to some major breakthroughs.
Okay, spill the tea.
Okay, so
Tell me about these breakthroughs.
for example, animal studies helped us develop shorter, more effective treatment regimens for drug-sensitive TB.
Okay.
They were instrumental in paving the way for using drugs like rifapentine and moxifloxacin,
Okay.
which have significantly reduced treatment time.
Wow, that's a game changer, especially for people who struggle to stick with the long and grueling, standard TB treatment.
Exactly.
So now what about vaccines?
Right.
We know that the BCG vaccine has had limited success,
Yeah,
especially in certain parts of the world.
for sure.
How are animal models helping us develop better ones?
Well, they're like the proving ground for new vaccine candidates.
Okay.
Scientists can test all sorts of vaccines in animals to see if they can train the immune system to recognize and fight off TB.
Okay, so our sources mentioned some promising candidates like Chedox 185A, MVA 85A, and the H56 vaccine.
Yes, those are good ones.
What makes these stand out?
So these are both subunit vaccines, meaning they contain specific parts of the TB bacteria that trigger a strong immune response.
Okay.
Chadox 185A MVA 85A uses a harmless virus as a delivery system.
Oh, so it's like a Trojan horse.
It is. It's like a Trojan horse.
Sneak attack on those TB bacteria.
Exactly.
I love it.
And then H56 is a fusion protein that combines several different TB antigens into one powerful package.
Wow.
Both of these vaccines have shown really great results in animal studies, reducing bacterial loads and sometimes even preventing the disease entirely. That's incredibly encouraging, but, science is rarely a straight line to success.
Unfortunately, you're right.
There must be some challenges in translating these successes from animals to humans.
Absolutely. And our sources provide a stark reminder of this with the MVA 85A vaccine.
Okay.
It looked incredibly promising in animal studies,
Okay.
showing great protection.
Yeah.
But when it was tested in humans,
Yeah.
it just didn't work.
Wow, that must have been such a disappointment.
Yeah, a huge disappointment.
Why do you think that happened?
Well, it highlights one of the fundamental limitations of animal models.
Okay.
No animal perfectly replicates human TB.
Right.
Our immune systems are complex, and the way the disease progresses can differ between species.
Of course.
So what works in a mouse might not necessarily work in a human.
So it's a reminder that animal models are just one piece of the puzzle.
Exactly.
And we need to be cautious about extrapolating results directly to humans.
For sure.
But despite these limitations, animal models have been absolutely instrumental in moving TB research forward.
They have been.
They've helped us unravel the secrets of this bacterial ninja.
They have.
And there's one more fascinating twist
Oh, yeah.
that came up in our research that I think is worth mentioning before we move on.
Okay.
And it relates to that concept of latent TB,
Right.
the ability of the bacteria to hide out in the body for years.
So like they're lying dormant, just waiting for the right moment to strike.
Yeah, exactly.
That's the traditional view of latency.
Okay.
But recent studies, particularly in mice, have challenged this idea
Okay.
and proposed a more dynamic understanding of latent TB infection.
A plot twist.
It is.
Tell me more about this dynamic hypothesis.
So it suggests that latency might not be a state of complete dormancy,
Okay.
but rather a constant low-level battle between the immune system and the bacteria.
Oh, wow.
It's like a slow, simmering conflict rather than a ceasefire.
So instead of taking a nap, the bacteria are constantly probing and testing our defenses. That's a whole new way of looking at it.
It is.
Wow.
And this dynamic hypothesis, if it proves to be true,
Yeah.
could have major implications for how we diagnose, treat, and prevent TB.
It's a reminder that even with a disease that's been studied for centuries, there are still mysteries to uncover.
Right.
And animal models can help us unlock those secrets.
Absolutely.
I'm already eager to hear more.
I know, me too.
But for now, we'll have to pause here.
Okay.
Join us for part 2, where we'll delve deeper into the world of TB research, exploring the challenges of drug resistance, the quest for better vaccines, and the incredible potential of new technologies.
Stay tuned.
Stay tuned.
Welcome back to our deep dive into the world of tuberculosis research, where we're exploring how animal models are helping us unravel the mysteries of this ancient and persistent disease.
Yeah, we've already covered a lot of ground, from mice to zebrafish, and how these different animal models have given us valuable insights into how TB develops, how it evades our immune system, and how it can linger in a latent state for years.
We also touched on the crucial role animal models play in developing new treatments and vaccines,
Right.
but not without some ethical considerations, of course.
Of course. So now I'm really curious to dive into some of the biggest challenges facing TB research today.
Yeah?
Our sources mention drug resistance as a major threat.
It's a significant hurdle. Drug resistance emerges when the TB bacteria evolve and become resistant to the very drugs we use to treat them.
Right.
This makes the disease incredibly difficult and sometimes even impossible to cure.
That's a scary thought.
Yeah.
And I've heard about these multi-drug resistant strains of TB.
Right.
It sounds terrifying.
It is concerning. Multi-drug resistant TB, or MDR TB, is resistant to at least 2 of our most powerful first-line drugs,
Right.
isoniazid and rifampin.
Okay.
And then there's extensively drug-resistant TB, or XDR TB,
Right.
which is resistant to even more drugs,
Oh, wow.
including some of our backup options.
So it's like these bacteria are becoming superbugs, evolving to outsmart our defenses.
They are. It's a constant arms race.
How do researchers even begin to tackle this problem?
That's where animal models become absolutely essential. They allow us to study how drug resistance develops in the first place.
Okay.
We can track the genetic changes that happen in the bacteria as they're exposed to different drugs
Right.
and see how those changes lead to resistance.
So it's like watching evolution in action, but at a microscopic level.
Precisely. And this knowledge is crucial for developing new drugs that can overcome these resistance mechanisms.
Mm-hmm.
We need to stay one step ahead.
So we can fight fire with fire, or in this case, superbug with superdrug.
Exactly. Animal models are also vital for testing new drug combinations against those tough drug-resistant TB strains.
So this is where those shorter treatment regimens we talked about earlier come in, right?
Yes, but drug-resistant TB is a different beast. The treatment regimens are much longer, often lasting 2 years or more,
Wow.
and involve a cocktail of different drugs, some of which can have harsh side effects.
That sounds incredibly grueling for patients, not to mention the strain on healthcare systems.
It is. And that's where animal models are contributing to finding solutions. They allow researchers to experiment with different drug combinations and dosages to see what works best against specific drug-resistant strains.
Okay.
They can also study how well these drugs reach different tissues and organs,
Right.
especially the lungs, where TB usually sets up shop.
Because a drug that works great in a lab dish might not be so effective in a complex living organism.
Exactly. It's about understanding how drugs work in the real world.
Right.
And this research is already leading to new treatment options for drug-resistant TB that are shorter, more effective, and less toxic.
That's really encouraging to hear. It offers hope to those battling these difficult-to-treat forms of TB.
It does. And it underscores why continued investment in TB research, particularly in animal models, is so crucial.
Right.
We need to stay ahead of the curve when it comes to drug resistance.
Now let's switch gears back to vaccines. We talked about how the current BCG vaccine has limitations,
Yeah.
and even those new vaccine candidates we discussed
Right.
face challenges in translating success from animals to humans.
Yes. That translation is a major hurdle. Our sources highlight several reasons for this.
Okay.
One is the sheer complexity of the human immune system.
Okay.
It's far more diverse and nuanced than the immune systems of most animals, making it difficult to predict how a vaccine will perform in humans based solely on animal data.
It's like trying to perfectly translate a complex novel into another language.
Yeah.
Some of the meaning is bound to get lost in the process.
That's a great analogy. Another challenge is the complexity of TB itself.
Right.
It has different stages of infection and interacts with the immune system in various ways.
We talked about latency, for example, and how TB bacteria can basically go into hiding for years.
Exactly. That makes it incredibly difficult to develop a vaccine that can target all stages of the disease and provide long-lasting protection.
So it's like we're trying to hit a moving target.
Right. And to make matters even more complicated, there's a lot of genetic diversity among TB bacteria strains.
Oh.
A vaccine that works against one strain might not be effective against another.
So it's a bit like trying to create a universal flu vaccine.
It is.
It's a monumental task.
It is. But despite all these challenges, scientists are making progress, and animal models are key players in this effort.
Okay, I'm all ears. How are animal models paving the way for vaccine breakthroughs?
Well, one way is by helping us understand the immune mechanisms that protect against TB.
Okay.
By studying how different vaccines work in animals, we can identify the key immune cells and molecules needed to control the infection.
So it's like reverse engineering the immune response?
Exactly. That knowledge can then be used to design more effective vaccines for humans. Animal models are also invaluable for testing different vaccine delivery methods.
Like those Trojan horse viral vectors we talked about?
Yes,
Really?
and also different routes of administration, like
Alters.
targeting the lungs directly.
Right.
By experimenting with these approaches in animals, we can optimize vaccine delivery for humans and potentially boost their effectiveness.
So even though there are obstacles, it's exciting to see that research in TB vaccines is forging ahead and animal models are paving the way.
They are. And now I want to touch on some of the remarkable new technologies that are revolutionizing TB research, many of which we've already mentioned.
Like genomics and those advanced imaging techniques.
Absolutely.
Yeah.
Those are transforming the field. But there are others as well. High-throughput screening, for example, allows scientists to rapidly test thousands, even millions, of potential drugs or vaccine candidates in a short amount of time.
Wow, it's like speed dating for TB research.
I love that. And it's significantly accelerating the pace of discovery. We're also seeing the rise of organ-on-a-chip technologies.
Organ on a chip. That sounds straight out of science fiction.
It does sound futuristic, right? These are tiny devices that mimic the functions of human organs, like the lungs.
Oh, wow.
They allow us to study TB infection in a more human-relevant setting without using live animals.
So it's like having a miniature human lung right there in the lab.
Precisely. And these organ-on-a-chip systems are already being used to test new TB drugs and study how the disease progresses.
Wow, it's incredible how technology is changing the landscape of TB research.
It is. And it's important to note that these new technologies are complementing animal models, not replacing them.
Right.
They're giving us more tools in our arsenal to fight TB
Yeah.
and helping us refine our use of animal models to make them even more predictive and relevant to human health.
So it's a win-win. More knowledge, better treatments, and hopefully one day a world free of TB.
That's the ultimate goal.
And we're back for the final part of our deep dive into animal models in TB research. It's been an incredible journey.
It has. We've covered a lot, from the basics of TB to the challenges of drug resistance and vaccine development.
Right, and those amazing advancements in technology that are changing everything.
Totally changing the game.
Now I'm ready to zoom in on the specific talents of each animal model.
Yeah, let's do it.
We've got this incredible lineup of creatures, each with its own strengths and weaknesses.
It's true. It's like a superhero team,
I like that.
each with their own special power.
Okay, so mice, as we've discussed, are like the workhorses of the lab.
The OGs of TB research.
Yeah.
They're small,
Yeah.
easy to manage, relatively inexpensive, and we can tweak their genes pretty easily.
Right, making them perfect for studying specific genes involved in TB.
Exactly. And because they have shorter lifespans, scientists can observe the entire course of infection in a shorter time frame.
That makes sense. But we also learned that mouse granulomas, those clusters of immune cells that try to contain the TB bacteria, don't perfectly mimic human ones.
Right. That's a key difference, and that's where guinea pigs come in.
Guinea pigs?
Yeah, they develop these necrotic granulomas that are much more similar to what we see in humans.
Okay.
They're also great models for studying how TB spreads, because they can be infected through the air, just like us.
So they're the transmission specialists.
Exactly.
What about rabbits? What makes them so special?
Rabbits are the go-to for studying cavitary disease. That's that severe form of TB where cavities form in the lungs.
Right, that sounds really serious.
It is. And they can also develop latent TB infection, which is tough to study in other animal models.
So they help us understand those hidden stages of TB lurking in the shadows.
Precisely.
And then we have the non-human primates, our closest relatives in the animal kingdom.
Right.
More expensive, ethically challenging to work with, but they provide data that's closest to humans.
Absolutely essential for studying the later stages of TB infection, how drug resistance emerges, and how effective vaccines might be.
Wow, they're like the elite squad,
They are.
bringing us the most human-relevant information.
They're key players for sure.
So it sounds like each animal model has its own specialty, its own expertise.
Totally agree.
It's like having a team of detectives,
Yeah,
each with their own skills working to solve the case of TB.
I love that analogy.
So speaking of research, our sources highlighted some fascinating studies I'd love to get into.
Yeah, let's do it.
One study from 2023 used mice to investigate the role of neutrophils in TB infection.
Oh yeah, neutrophils.
Heh.
Those are our frontline soldiers against infection.
Right, but in this study, researchers found that neutrophils can actually worsen TB disease in mice.
That's right. It was a surprising finding.
Our own immune system is making things worse.
Well, in a way, yes. They found that neutrophils contribute to inflammation in the lungs, and that inflammation can actually damage lung tissue, making it easier for the bacteria to spread.
So it's like the immune system is overreacting and the friendly fire is hurting the host.
Exactly. It's a delicate balance.
That's a really important finding. It opens up new possibilities for treatment.
It does. Imagine if we could develop therapies that dampen that neutrophil activity and prevent that lung damage.
That would be amazing.
It'd be a game changer.
Our sources also mentioned a study on leprosy, which is closely related to TB.
Right, leprosy is caused by a different bacterium, Mycobacterium leprae, but it also forms granulomas and can cause serious tissue damage, especially to the skin and nerves.
And this study focused on the role of lipid droplets in leprosy.
Yes, lipid droplets are essentially tiny fat globules found inside cells.
Okay, I never would have guessed that fat would play a role in this.
It's surprising, right? This study used a combination of cell cultures and animal models to figure out how these lipid droplets influence the immune response to leprosy.
So what did they find?
Turns out leprosy bacteria can actually steal lipid droplets from host cells.
They steal them.
They do. They use them for their own benefit.
How does that help them?
Well, the lipid droplets can actually interfere with the ability of immune cells to kill the bacteria.
So they're using our own fat against us.
In a way, yes. They can also boost the production of anti-inflammatory molecules which suppress the immune response.
Wow, those sneaky little bacteria.
They are incredibly adaptable.
So what are the implications of this for treatment?
This research opens up the possibility of targeting lipid droplets as a new way to treat leprosy and maybe even other mycobacterial diseases like TB.
That's really promising.
If we can disrupt this process of bacterial hijacking, we might be able to boost the immune system's ability to fight off the infection.
I'm also really intrigued by the human challenge studies that our sources discussed. It's amazing and a little unnerving to think about healthy volunteers being intentionally infected with a weakened form of TB.
It's definitely a complex ethical issue. But these studies give us invaluable data that we just can't get any other way.
Right.
We can directly observe how the human body responds to infection and how well potential vaccines work.
One study in South Africa used this model to test a vaccine candidate called MVA-85A,
Yeah, MVA-85A.
which had shown great promise in animal studies.
It did look very promising.
But unfortunately, it didn't work as well in humans.
No, it didn't. It failed to provide significant protection.
That must have been a huge disappointment.
It was a major setback, but it highlights how important rigorous testing in humans is.
It's a good reminder that science is a process.
It is. It's full of twists and turns.
Sometimes we make huge leaps forward and sometimes we hit roadblocks.
That's the nature of research.
And each study, whether it's successful or not, adds to our understanding.
Exactly. We
Yeah.
learn from every experiment.
Our sources also described a groundbreaking development that could change how we do these human challenge studies in the future.
Oh, yes. The triple kill switch strain.
Triple kill switch. That sounds intense. What is that?
So scientists have engineered a new strain of TB bacteria.
Okay.
It has 3 built-in safety mechanisms that
Wow.
allow us to quickly and easily eliminate it if we need to. It's essentially a self-destruct button for the bacteria.
That's amazing. So it's like a safety net for the volunteers in these studies.
Exactly. It could make human challenge studies much safer, allowing us to test new vaccines and treatments in a more controlled and ethical way.
That's incredible. So as we wrap up this deep dive, I have one final question for you and for our listeners.
Okay, I'm ready.
With the rise of drug-resistant TB, the need for better vaccines, and the rapid pace of technological advancement, what role do you see for animal models in the future of TB research?
That's a great question. I think animal models will remain absolutely crucial. They help us understand the complexities of TB tests, new treatments, and vaccines, and ultimately reach our goal of eradicating this disease.
It's a global problem.
It is. And we need every tool we can get. But it's essential that we prioritize responsible and ethical research practices.
Right, minimizing the number of animals used and making sure they're treated humanely.
Absolutely. It's about finding that balance.
Using these models to gain vital knowledge while respecting the well-being of the animal.
Exactly. And remember, animal models are just one piece of the puzzle.
Right.
We also need human studies, clinical trials, and epidemiological research to develop effective ways to control and eliminate TB.
It's a team effort.
It is a global effort.
Well, thank you so much for joining us on this deep dive into the world of animal models and TB research. We hope you found it fascinating.
Thanks for having me.